Eric Randolph. Eric worked at the EPA for about 3 years, right, before he came to start our PhD program the fall of 2017. I am his advisor, and today he's going to talk to us about a new learning paradigm in aplysia that he has mastered and brought a lot of us along with him in the lab, and give you some of his initial data in using this learning paradigm and what it might be able to do for us. So take it away, Eric.
Thank you so much for the introduction. Hey everyone, my name is Eric Randolph. As I said, I'm a second year PhD student here at University of Erasmus. Today I want to talk to you about the charismatic creature I've been working with. I call him the learning slug. His real name is Aplysia californica. So for this talk, I'd like to go over a little outline, some background on some classical forms of memory, go over some physical and molecular changes that happen that help us encode for these memories, and then talk about a gene that I'm interested in. It's called terneurin, which I believe helps make these memories. And then I'll go over some methods. I'm gonna go over the behavioral protocol, the one that I did for these animals. I'm gonna go over the analysis that I did for that, some results in the gene expression of terneurin, some conclusions and future directions. So without that, there are two forms of memory that we are interested in: is long-term memory and short-term memory. And while this depiction looks a little busy, I'm gonna walk through it. So for short-term memory, we have a neuron giving a weak signal to another neuron, and the weak signal just results in a greater release of neurotransmitter into the synaptic area, which responds in a greater response on the receptor cell. Whereas long-term memory results from a stronger stimulus coming into the neuron, and actually the neuron will then create new proteins, which will go to the synaptic cleft area, and it will change the size or strengthen the synapse area of the neuron. Demonstration of these morphological changes. If you look here, this is actually a neuron from a Drosophila, and through everyday experiences, the neuron will morph and change and grow these axonal processes called dendrites. And it is these dendrites which can connect to other neurons and make more synapses. And it's this morphological change that encodes the experience, or what we consider as memory. The gene I'm interested in is terneurin, and terneurin actually helps make these morphological changes. When it is expressed, it has a role in reorganizing the actin tubules within inside the nervous system, and it will start to grow these new dendrites, or grow extensions off of dendrites. Terneurin is a highly conserved gene throughout the animal kingdom. It has been studied in mammals, birds, Drosophila, and C. elegans. And so I want to see if I could take this terneurin gene and see if I can use it as a molecular indicator for the formation of new memories. In order to do that, I thought of a way: how could we follow a memory? How could we teach an animal? So we decided to follow Saswine feeding protocol. Saswine is a researcher who came visit Erasmus about a year ago and taught us this protocol. And the protocol, it helps an animal to learn that an inedible food source, recognize it as being inedible. And the food source we chose was Ovolactuca, which is a food source that the animal was raised on, among others. And we wrapped it inside 75 micron Nitex netting to create these probes that the animal could smell and taste the food, but was unable to ingest it, so it was not able to swallow it and get the nutritional benefits of the food. Okay, so this is just an example of the normal biting reflex of the aplysia. The cortical cavity showing this regimen of this feeding. So the behavior protocol is broken up into two times. So there's an initial training period, which we call day 1 training, and a retraining period, which we call day 2 training. And in day 1 training, we fast the animals for 48 hours, and then we present the food probe. The animal then goes for the probe. We calculate and keep track of the total time that the animal keeps it in its mouth. Normal behavior for these animals, they'll go for the probe, and they'll spit the probe out because they cannot swallow it, and then they'll go for it again multiple times. And then after 3 minutes of the last time the animal spit out the probe, we will call day 1 training complete. We'll then tally up the amount of total time within those intermittent periods of the animal keeping it in its mouth. If the total time within the animal's mouth that it held the probe is greater than 100 seconds, that animal is a good candidate for day 2 training. If the total time is less than 100 seconds, then the animal is considered a dud, and we do not use it in further analysis. So 26 hours later, we bring the animal back. The animal is then again presented the food probe. The animal goes for the probe. We again calculate the total time the probe is held in the mouth, and we then determine and see, is the total time held in the mouth on day 2 less than it was on day 1? If it is, then we use that as a proxy for learning that the animal was able to recognize and remember this probe from the previous day. At which time we then dissect the buccal ganglia, which is this one right here, and we run a qPCR on it. We chose the buccal ganglia because it is the one that controls the buccal mass, which is responsible for the feeding, which is what you saw in the video of the opening and closing. So if the memory was going to be held somewhere, we hypothesized that it would be held in the buccal ganglia. One more thing, when we were raising these animals, they were raised in an exercise regime. These were all sibling animals, so they're of a single cohort. We raised them in these tanks. We call it a dump bucket setup, but they actually in the aquaria. These buckets fill up with water, and they're about once every 5 to 7 minutes, and then dump it to their aquaria. So this is a still water, but then it gives them this turbulence, and this turbulence is supposed to have them exercise, so they'll have to grip on with their foot onto the wall to not get washed away. We did it this way because last year we did a pilot study of this, and we had animals raised in still water as well as dump bucket water, and we found that the animals raised in the dump bucket seemed to complete the behavioral protocol and learn better than the animals raised in still water. So let's look at some results. So right here we're going to look at the percent savings. This is the time that the animal kept the probe in its mouth in day 2 versus the time, or divided by the time in day 1. So it's saying, did the animal recognize the probe faster? How quicker did the animal recognize that the probe was inedible on day 2 versus day 1? And we see there's around a 64% savings. So the animal on day 2 recognized the probe almost half as fast than the animals on day 1. So we consider that that's pretty good. So the animals, they learned that this probe was inedible. And then so we went to gene expression. So if we look at our gene expression results, we see that trained animals on the right side, there were 13 animals we chose from the previous 14 animals on the previous slide versus 13 animals that had never seen the probe at all that were our controls. And we see a slightly higher Terneurin expression between the two. So what does that all mean? So in conclusion, we would say that only through long hours, hard work, and determination, and the tireless commitment of these 3 wonderful undergrads, I was successfully able to reproduce this behavior, this protocol, and get my pleas to learn. This learning was demonstrated through the total time in day 2 in the mouth being less than the total time in the mouth on day 1. And it kind of suggests that learning may be linked to the increased expression of Terneurin. That leads me to some future directions in this research. We're wondering, the future directions will address the question that could the Terneurin expression in the target cells that we're looking for be obscured by too many surrounding cells? This is what half of a whole buccal ganglia looks like. We use whole buccal ganglia for this, but the neurons that we believe or through literature says that is really responsible for the buccal mass movement are the neuron B31 and B32, right here in red. And if we're getting the entire buccal ganglia, we feel that all of these other neurons that are expressing Tunerin as well could be obscuring the elevated expression of just the two neurons that are responsible for the action. So we want to try to do a targeted approach of getting just these two neurons, or less than those two, less than the whole buccal ganglia. And this is an image under the microscope where these two neurons can be really hard to find. We are able to find these two neurons, which are this one here, B2 and B1. It'd be like landmark neurons. And so we're actually able to like cut the buccal ganglia maybe in half and just get this section of it. We'll be sure to include B31 and B32. We will get rid of a lot of these sensory neurons and extraneous motor neurons to get a better precision of our expression. And one more future direction I would like to look into is looking at the difference between Tunerin and its exon terminus of TCAP. Tunerin is a large intermembrane protein, right here stained in red, and it stays on the inside of the membrane of the cell, where TCAP actually goes into the cell, into the cytosol. And since we know the function of Tunerin, how it changes the actin into the cytoskeleton, I imagine that perhaps it's the TCAP inside that's changing that. TCAP can be formed in two ways. It can be a peptide cleavage or a splice variant. And if it's a splice variant, then I'm worried that if we're looking for the whole gene Tunerin, we're missing the mRNA for TCAP. So TCAP specific primers are being designed right now to see if I can see a higher elevation in TCAP, because maybe that's the one that's being highly expressed or upregulated during learning instead of Tunerin. All right, with that, I'd like to give some acknowledgments. I'd like to thank first and foremost my advisor, Lynn Fever, for helping me through this. My lab mate, Nick Crom, who helped me from beating my head against the computer many times. My great undergrads, Mia DiCaprio and Faith Bellas from University of Miami here in Coral Gables, and Cassandra Schultz, who did a summer intern from University of Tulane in New Orleans. Also thank the funding, NIH, and special thanks to Athleisure Resource Facility, who helped, who keeps us going with these animals. And with that, I'll take any questions. Yes.
I wonder if it would be interesting to look at Tunerin expression in your learners compared to the athlesia that you kicked out of the experiment because they couldn't learn.
It would. We actually did the extractions on the duds, so we are going to test that too. That would be interesting. I agree. It's in the process. Yes.
In the graph showing the difference between the Tunerin expression in the untrained versus trained animals, was that different? Was that a significant
That was
difference?
not
or just
significant.
yours?
It was a higher expression, but it wasn't significant.
So you do the first trial in one day, and then you do the second, the T2 is the second day, is that right? The very next day?
Yes, 26 hours later.
26 hours is what you said. Is there a specific reason behind the timing of that? Is that just from the protocol, or is that based on their like known memory span?
That was based on the protocol, but it was done so that the animals can have time to develop these new synapses and think about what happened the first day.
Have you played around with shorter or longer times?
I have not.
Can you say why?
Because
Why would shorter not work?
shorter wouldn't work because I don't know, wouldn't be enough time. I just don't think it would be enough time for the genes to act in the way it would. Yes.
Can you look at like IHC or inside tubes I don't know, it seems pretty hard. So
I'm sorry.
the microdissection seems sort of hard. I was wondering about inside tubes or immunohistochemistry to see if those two cells actually have higher protein or expression levels.
Situ. I don't know how it would do in situ. I would have to look into that more so. I have to do in situ studies. Yeah, because basically when you pull out the ganglia, that's it. So I don't know another way to visualize it.
Antibodies.
Huh? Antibodies? Oh, okay. I was told that antibodies are really hard to make. So but yes, it is something to look into.
Any clue on why learning may be mediated by the conditions of the water? You know, the fact that you could actually need, you said that you put excess flow or something.
So
Why is
for the
that?
exercise,
Yeah, and exercise.
I don't know, I'm under the impression of kind of like how things learn better when they're exercise, or like how children at school, you know, you give them recess so they can like learn more and stuff. So maybe that's a process. If somebody's just sitting around not doing anything, it might not want to expend energy to learn as well as if something was busy or moving around.
Thank you, Eric.